So, as I mentioned earlier, our next speaker is going to present about his research and data on lipid-based formulation development. Obviously, that's another technology that is highly useful and helpful and has been used for many decades in bioavailability enhancement. So our speaker is Dr. Yogesh Bachhav. He is a pharmacist by training, has a PhD in advanced drug delivery systems from ICT in Mumbai, India. He has around 16 years of post-PhD experience in Europe in the field of pharmaceutical development and development of investigational drugs. He has contributed to success of clinical candidates ranging from preclinical to phase 1, phase 2, phase 3, and commercial. Yogesh has worked as a research scientist for around 4 years and on a collaborative project between Pantech Biosolutions AG and University of Geneva. And he is currently working as Associate Director at AiCuris Anti-Infective Cures AG in Germany and responsible for pharmaceutical development and investigational drugs in the domain of innovative and antiviral and antibacterial drugs. So let's hear from Dr. Bachhav about lipid-based formulation development and his experience and insights on that. Welcome.
Good morning, all. So glad to see you had a nice coffee and some food, so my job becomes a bit easy. I don't have to spend some more efforts to keep you awake. So I think from yesterday we have heard a lot regarding the lipid-based formulations to maximize the bioavailability of the poorly soluble drugs. So maybe some of the topics are a bit redundant or they're already discussed, so I'll try to pass a bit faster for those topics there, and the others I can talk. Yeah. Regarding the contents of my talk today, so high-level overview of lipid-based formulation, physicochemical and physiological aspects for these formulations, what are the steps normally involved to develop lipid-based formulation, then actual formulation aspects, then we can jump to some prototype technologies, case studies, and the current trends. So why one should think of lipid-based formulation when it comes to the drug discovery? I mean, as Firoz said, most of my experience comes from the drug discovery companies, starting from really the lead candidate selection, followed by the preclinical and the early clinical research. At that point, it's very important to decide, and typically when I work with the oncology drugs or the drugs which are active in the field of antiviral segments. So these drugs are quite poorly water soluble, often they are quite lipophilic. So the question is then, which technology is best suited to increase the bioavailability? So when we look at lipid-based formulations, so in this case, we have chance to solubilize the drugs having the high log-P. There's also a chance to decrease the efflux by transporters. We had a chance to work with the cyclophilin inhibitor where we had a PGP efflux. There is also a chance to increase the permeability, typically if you have a BCS class 4 drug. And then sometimes being anti-infectives or from oncology category, maybe you want to target the lymphatic route, and if you want to rule out the first pass metabolism. So all these factors can help to select the lipid-based drug delivery system for the further development. How do they work basically? So this understanding of the digestion of the lipids in the GI tract is very, very important. The reason I want to tell, once we had a drug which was being developed for HIV indication, And we tried solid dispersion by hot melt extrusion. We tried nanosuspension. We tried using the acids because it was slightly basic drug. Nothing was helping, and I don't want to endorse Gatafos here, but we simply use the drug dispersed in labraphil lipophile drug without any solubilization, and the increase in bioavailability was massive. I mean, then somebody might think, oh, drug was not even soluble in that lipid. How it may help to increase the bioavailability? So understanding of this digestion pattern of these lipids might be of help, because in the presence of the gastric enzymes, these triglycerides are getting digested into the monoglycerides, followed by the micelle formation. And this in vivo formation of micelles is very important often to increase the bioavailability. So sometimes you may not even see the solubility of the drug into the given lipid, but there are still good chances of having the higher bioavailability, and that's where this digestion of lipids in vivo is very important. So what should be the considerations we should take into account when we try to develop the lipid-based drug delivery systems? So when it comes to the physical aspects, so what are the solubilization potential options we have of different lipids of a given drug? So there we try to select the range of lipids offered by different companies like Gatafos. Maybe you have other abitic possibilities, medium chain triglycerides, long chain triglycerides. Often the solubility in LCTs is not so great. Maybe MCTs are of help there. So there we need to check what is the solvent capacity of these lipids, what are the typical droplet size we are able to get once they are emulsified. Once the lipid-based system is dispersed, what kind of globules you are able to get. And of course, hydrophobicity of the API is also of relevance, because often we talk about the lipophilicity, but we have to also check whether it has any affinity towards the given lipid, and that is also something quite important to understand. Chemical stability aspects are also quite important, because often some of the APIs, they are not quite stable into the given lipids. So the drug excipient compatibility data at early stage is also important. I can cite one more example. We were developing a cream formulation for antiviral drug for topical application, working quite well in terms of the skin permeation, skin deposition. But over 6 months storage at 4075, we saw that one of the oxidative degradation product was going out of specifications. We tried to understand a bit more, and that was linked to use of the PEG. Of course, PEG is not a lipid excipient, but it has some residual oxidizing impurities, right? Then we just thought there are super refined PEG options available for some of the specialty excipient companies. We tried to replace this normal PEG with the super refined PEG, and surprisingly, data was quite good. Of course, when you go with this data to FDA, questions will be certainly asked. Why you want to use super refined PEG? Why not a normal PEG? And this data is what we have generated at that point. The stress stability data or the IC stability data was of help. Of course, then it's other discussion of cost of goods. When you go for out licensing or scale up, you really want to use this cost expensive super refined PEG, or maybe good idea to change this PEG with some other excipients, or at least maybe addition of antioxidant. But there also you need a rational, the moment you want to add preservative or antioxidant to the formulation, you need to have sound data or rational to justify the addition of excipients. But what I'm trying to say, having the data on the drug excipient compatibility data, and if there is any sensitivity of the drug towards the residual oxidizing impurities, which is often the case in the lipid excipient, that should be taken into account. And physiological aspects are also important. The digestibility option I already discussed in the beginning, how the given MCT or LCT is getting digested in vivo. That is something important to understand. Whether the given lipid is going to interact with the efflux inhibitors. Often we have the option of using this efflux inhibitors, like for example, poloxamine, this polymeric surfactant we use to interact with the efflux inhibitor in case you want to decrease that efflux and increase the bioavailability. This is something important. Several lipids have this property to interact with the efflux inhibitors. That is something to be carefully checked. And then having said that, the drugs molecules having Griswold like properties or brick dust compounds, they are both suited to be incorporated into the lipid-based drug delivery systems. So having some solubility screening interaction of these APIs in the given lipid matrix is really important to understand if these systems can be used to be developed as the lipid-based drug delivery systems. So what are the typical steps involved in developing the successful lipid-based formulation? And then we can come with typical lipid-based drug delivery systems. It could be microemulsions, nanoemulsion, and these are originating mainly for SAIDs or SMIDs as long as they are not diluted with water. Then you have the options of developing solid lipid nanoparticles, NLCs typically for the oligonucleotides or maybe siRNA. These delivery systems are quite popular these days. So first step, we try to determine the solubility of the drug into these lipids. Next step is to emulsify in the given surfactant by often the co-surfactant is added, and then performing this ternary phase diagrams is quite important because there you want to really see in which particular region my microemulsion region remains. That really helps you to optimize the concentration of oil surfactant and co-surfactant. One of the advantage of this ternary diagrams, I can go back. To go back. Sorry. Yeah, thanks. So we have reported one article where it was possible without using any polymeric agents, just by varying the concentration of oil and surfactant and co-solvent, it was possible to get the semi-solid microemulsions. So you get typical gel-like concentrations. So sometimes maybe then this information is quite helpful in case if you are developing a IV formulation where you don't want to have any sol to gel transition. So this thorough optimization of the given system using phase diagram is very, very important. In terms of characterization, evaluation, then certainly you want to understand the globule size, polydispersity index, zeta potential, whether your formulation is stable to the freeze-thaw cycling, and whether there are any long-term stability issues. This is something important. And then digestion is also something important. There we have the in vitro lipolysis test. That really helps to understand, without going for in vivo studies, how the given system is going to behave when it is given by the oral route. Then next step comes often for, we have heard yesterday presentation from GLAD and other speakers. So converting this liquid formulation into the solids is also important. Often these liquid formulations are not suited due to the leakage issues in the hard gel or soft gel capsules. So maybe sometimes there's a demand from the commercial team or marketing team, if it is possible to convert this liquid formulation into the solids. We learned yesterday there are different processes possible. There are different carriers option possible from ranging from MCCs to silicas. We can go for different methods listed here. Simple blending followed by the melt granulation. Supercritical fluid extraction is also something quite important where the CO2 can be used. But let's not make it so complex. We try to keep it as simple as possible when it comes to the drug discovery. But what I'm trying to say, if the need arises, if we have to absorb the SMIDs formation, the SAIDs formation onto the solid matrix, then we can think of using these carriers by using the commercially feasible process in order to get the free-flowing powder, which can be compressed into the tablets or can be taken as a capsule. One of the example we can cite is the GenGraph, where they have tried to convert the sets or formulation of cyclosporine into the solid capsule, the oral solid dosage form, because new oral or Sandiimune was the, let's say, the traditional or conventional formulation of cyclosporine, and they have this new version GenGraph. And then I come to some of the platform technologies based on the lipid-based principle. The first is Lidose technology, which is developed by SMB group. Basically, Cal4 and SMB technologies are involved. So what they do basically, they use the excipient like Gelushire, and they also have a stabilizer like cellulose. So basically they just go with the melting of this semi-solid excipient like Gelushire, and drug is simply dispersed there, and the polymer like MCC takes part as a stabilizer in that system. And the advantages of these systems are there is increase in absorption. The API is also protected against the oxidation, and there is increase in the bioavailability. There is no food effect, and due to that absence of food effect, the inter-individual variability is also reduced. And this technology they have successfully used for the low dose substances, the drugs which are unstable to light, air or humidity, and slightly soluble drugs. So principle remains quite simple. The excipient is molten. Like Gelushire is like, if you see typically they have the melting point in the range of 20 to 25 depending on the HLB values they have. So the excipient is molten, the drug is simply added there and then it is solidified, and the stabilizer we are adding it ensures that drug remains dispersed into the given formulation. Typical data I was able to get, this was for fenofibrate, where they tried to compare the dissolution profile and even the PK data in vivo for the formulation developed by using Lidose technology against the nanoparticle formulation, and you can clearly see there was difference of almost 58% of the bioavailability compared to the nanoparticular formulation, and the food effect was nullified using the Lidose technology, which was the major drawback of the nano-based formulation. So alone particle size reduction may not be of help. Sometimes we might have to take the advantage of this lipid-based technology to get rid of this food effect for the poorly soluble drugs. Then another interesting technology is from Microgics. This is developed by Galenix Innovation. In this case, they try to use the carriers like maltodextrin. So what they do basically, the microemulsions are adsorbed basically, and these are the microemulsions are not containing any solubilized drug. So basically it is a suspended drug. So microemulsion suspensions, they are adsorbed onto the maltodextrin, and then this can be used to enhance the bioavailability. This approach can also be used to increase the stability of the potential molecules which are not quite stable. And this technology can also be used for the water-soluble drugs like proteins, because in this case what you do basically, you dissolve the drug into the aquasphere, you go with the W by O microemulsion, and that is being adsorbed onto the carriers like maltodextrin, and this technology can work. And the development pipeline they are working is for lansoprazole, fenofibrate, esomeprazole, and valacyclovir. It works quite well. You can also see the dissolution profile for the two drugs there. So, sorry again. Can you go back to previous slide? Yeah, so you can see the dissolution for the drug progesterone with the given technology. So there's a clear evidence of increase in bioavailability compared to the conventional formulation. It works quite well. And this is one important platform technology from Lonza. This is a melt spray chondrial process. And here what they do basically, they can even use this technology for hydrophilic drugs. So hydrophilic drug is suspended into the molten molten lipid matrices. So basically you get the lipid multiparticles technology. The advantages of this technology, again, you have the self-emulsification because of the presence of the lipids. Extended release is possible because often we use the lipid-based formulation to have the immediate release. But imagine if you have the highly water-soluble drugs where there's an initial burst which may lead to some adverse event or undesired side effects, and maybe there's a rationale to prolong or to have the sustained release. So that advantage can be achieved by using this technology. So you can get extended release properties. The excipients they are using, they are the grass excipients, so there is no excipient related adverse event really. Test masking is something important typically for the drug like azithromycin or clarithromycin, which are being used into the pediatric population or geriatric populations where the test could be a major issue. So in this case, that's advantage. Then this technology can be used to disperse the drug into the lipid excipients, and the test masking could be achieved. So here is that example where they tried to utilize this technology for azithromycin. So it was possible, you can see from the graph, compared to the sachet product, with using this LMP product, it was possible to reduce the Cmax and to have the extended release. And along with that, there was also advantage to have the palatability because the drug was coated by using the lipidic matrix. Neural is something quite interesting. The reason I'm going to cite here, because yesterday also it was quite significantly discussed, but in my previous job we had one cyclophilic inhibitor which was in phase 3, later it was out licensed to Novartis, and it was having from the same class, and there we had a quite deep dive into the neural formulations because the molecules were quite closely related, having the same log phase, same structure. So basically neural formulation, if you see the first generation, it was Immune, where it was a corn oil based solution, and there was quite food dependence, and there was incidence of variable bioavailability ranging from almost 10 to 89%. So what they did basically, they just replaced the corn oil by different oil, and they came with this SEDS oral formulation, and this had a result of minimized food effect, and they could get consistent absorption, and this inter-individual variability in terms of the bioavailability was reduced, and this was something quite important basically. So what we did basically then, maybe the next case study will show that what we did with our own molecule, but again this is something quite important, maybe Firoz will like that, because I remember when you came to us and you wanted to highlight this microemulsion approach, and this was the task given to me, because when you are into drug discovery, the amount of molecule you have might not be quite significant. You can really explore a lot of options. So what we try to investigate whether it's possible to use this microformulation approach to at least to screen in which kind of oils or surfactants or co-solvents we have good solubility. So basically we try to use this 96 well plate to prepare the drug solutions. Solvent was evaporated, so we have microgram quantities dispersed into each well. Then the vehicle was added. We subjected to agitation at 48 hours at room temperature, followed by filtration or centrifugation, and we were able to get the solubility data. And that was typical graph we had, and it was possible to screen like 20 to 40 compounds in a series. We hardly needed 20 milligram of compound for the screening. And based on this data, at least we were able to investigate the druggability aspects. Imagine we had like one lead compound and other backup compounds, and the question is asked to the CMC team, okay, which of the compound from the given series has a good probability of success in order to formulate. This data was quite important, and I mean it was saving some time. We don't need a lot of drug, and this was quite helpful at that point. And using this concept or this microformulation approach, we were able to propose few formulation for the quick PK studies or tox studies. Sometimes the simple suspension formulation might not work for the tox studies because of you get plateau effect, and then of course no matter how much drug you are dosing, there is no increase in bioavailability. Certainly you need an enabled formulation like lipid-based formulation, and this was something quite important there. And this was the case study I was referring in relation to the neural. So we had this molecule in phase 3 trial, and this was being developed against hepatitis C. So structurally it was quite similar to cyclosporine, and then we wanted to investigate the existing neural formulation there, and the patent was also referred. But in that case, we could find out, in contrary to the prior art, the presence of water component into the formulation was very, very important. So it's not just mixture of oil, surfactant or co-surfactant which was influencing the solubility. So into the final formulation, the presence of water component in the range of 15 to 20% is quite important to have the given solubility, and this was systematically investigated. Other case study, this molecule was being developed as an anti-diabetic molecule, My colleagues tried to use it by using simple suspension formulation. Again, at early stage, the synthetic process is not quite optimized. It's quite common you get the different polymorphic forms, different particle sizes, and when you run different PK studies, safety studies, or tox studies, the exposure might be different. So there we simply tried to come with the lipid-based formulation, and as evident, using lipid-based formulation, the PK data rats was quite there was significant increase in the bioavailability, and using this approach, it was possible to go to the next level of the preclinical studies. This was another interesting case studies basically. So we had in license one compound as a TK inhibitor or the oncology indications. As per my pharmacologist, this drug was a bomb, but with the given suspension formulation, when we ran some efficacy studies, we saw no activity, and then discussion was why we are not able to see any activity or any pharmacological activity into the given animal species, and we realized the formulation, the simple suspension formulation was not homogeneous, drug was not really, we were not able to deliver the drug in the desired concentration, and then we came up with first homogeneous suspension that looked quite good, and there we saw less variability between the different animals, but again the exposure was not good enough. There was less, there was also impact of the polymorphism, and finally we came with the lipid-based formulation. These are the photos from the lab batches, of course, at that time. And using this lipid-based formulation, it was possible to rule out the polymorphism, but at the same time the bioavailability was quite good, and it was possible to correlate the pharmacological activity as it was reported, and then it helped us to go to the phase 1 based on this data. And this, the same cyclic peptide I discussed before, because it was at phase 3 already, and then we wanted to come with the commercial formulation. So the formulation was soft gel capsule, and then final formulation has a 20% ethanol almost. Yeah? And this was putting a lot of burden on the process because of the presence of the ethanol. And then question was whether it's possible to get rid of ethanol. So what we did, we ran some design of experiment studies. The typical variables we studied, drug loading, oil content, surfactant content, co-surfactant contents, and what we checked as an effect is a particle size upon tenfold dilution in passive, and the dissolution data. We tried to understand what impact it has if we try to change the different factors listed here. And what we observed when we tried to change the concentration of ethanol, initially it was 20%, so we tried to bring it down to 10%, 5%, even 0%. Of course, on the other side, then we had to play around the concentration of lipids and the surfactants we were using. And at the end, there was not much impact onto the overall particle size upon dilution, upon the dissolution, because presence of the lipids and other surfactants were able to take care of that final particle size as well as the dissolution profile. As drug was basis class 2, so based on the dissolution data, it was possible to justify that removal of ethanol. It was possible to achieve the same dissolution profile than before, and based on this data, then we tried to get rid of ethanol from the final formulation. And with that, then I would like to add a few thoughts on the current trends. So the supersaturable SMEDs was something quite relevant, but this is already discussed quite a lot since yesterday, so I will not talk a lot. But again, the combination of solid dispersion and this lipid-based formation could be something quite interesting, because we have the possibility to achieve the supersaturation state. Combination of the LCTs and MCTs could be relevant, because often during excipient screening, what we do, we try to run the saturation solubility into the individual lipids. Maybe good idea to also screen for the mixture of the lipids. This is something I also seen, because often we go to the IgG limits, any lipid is not allowed beyond some concentration. Maybe to bring down that concentration, we try to compensate by addition of any additional lipid. So in that process, what we have found into the mixture of two lipids, maybe the solubility of the drug is quite different compared to the single lipid, and that advantage can be taken. And at the same time, we get advantage of this IgG limit that we still try to be within limits. Then we also have the possibility of combining lipid carriers with the polymers. So basically we go with this lipid polymer hybrid nanoparticles. This is something quite interesting platform technology is coming. Then AI-based formulation predictions could be of importance when we go for personalized medicines there. And I think in our previous talk we also saw Shaukat briefly mention use of the lipids could come with the long-acting parenterals, because often for depot formulation we try to consider nanosuspension or hot melt extrusion, but lipid-based formulation can also be explored for the other depot formulations. So to conclude then, the lipid-based formulations, they have evolved into a proven and commercial validated platform, as we see from the example of cyclosporine. And there is a huge potential to increase the oral bioavailability of the poorly soluble drugs. We have the advantage of reducing the or avoiding the first pass metabolism. We also have access to the lymphatic transport, and as an end result, we have the highest systemic exposure. There are breakthrough technologies like neural or lipofan, and they have already demonstrated that this technology works quite well to increase the bioavailability. The case studies which I showed, so there were some innovative application of lipid-based formulation to increase the bioavailability of challenging molecules or difficult to formulate molecules, because these were the points where clear go or no go, because you are standing at the position, you need to tell your management whether this molecule is going to work in the given indication or not. And CMC could also be one of the criteria, or drugability is also one of the criteria they do consider before the molecule is in license or they want to take it for further development. And then as we discuss the recent trends, so there is industry shift where we have to consider these new technologies like supersaturation or use of lipid polymer hybrid models to develop the lipid-based formulations. With that, I would like to thank you, and if you have any questions.
Hi, thank you for your talk. So I'm kind of curious. So if you look at the last 5 years, if a drug has solubility issue, most likely people use the amorphous, either hot melting or spray drying. And even you look at the history of lipid formation, there's not much use of that. I'm just curious, what's the reason why people didn't use, at least all the way to commercial?
Sure. I mean, this was also discussed in some of the previous talks. So the chances of commercial success are a bit higher when it goes for the use of amorphous solid dispersion. The reason is that the polymers you are using in amorphous solid dispersion, the typical safety concerns are not that big when it comes to the lipid. Then it is possible to convert this amorphous solid dispersion into the oral solids quite easily compared to the lipid-based formulation. So there are several factors you can consider why then trend is more towards amorphous solid dispersion compared to the lipid-based formulations.
Yeah, also one correction. I think you have one slide about neutropene as a long-acting injectable. It's not lipid. It's actually a POJ microsphere.
Thanks.
I think also if you look at the tablet press versus soft gel, I don't know, have you seen soft gel machines, how they run? The speed-wise?
You can solidify, right? You can solidify.
I know, but it's still, the speeds are significant, and the solidification is being done. People are using lipids in amorphous solid dispersion. But when you look at tablet press, millions of tablets in an hour versus maybe 100,000, 200,000 soft gels per hour. So oftentimes I have seen tendency to go towards technology that can pump in millions versus hundreds of thousands. So it is not just the technology not being able to do it, but also, in my view, there is also the economy
one more thing I can highlight you. Imagine I have a drug for tox studies. There are two options, amorphous solid dispersion lipids. The polymers I'm using, they are not getting absorbed. So whether I'm using quite higher concentration, my toxicologics is not concerned. But at the same time, I'm using quite higher concentration of lipids. Naturally, I'm going to see some side effects, and then I need to justify whether it's really needed. So that really depends on case-to-case basis rather than making it quite general, I'll say. Yeah, of course. Exactly, that's what I'm coming from. For tox, you have 10 times higher dose compared to your clinical dose. And can you imagine that the quantity of lipid you might need for that dose? Then certainly it's no go. Then you have solid dispersion, 30 to 40% drug loading. So maybe then you have better chances of success when you go for amorphous solid dispersion.
Yeah, to prove my point about tablets, why